Hello to everyone and welcome to this virtual session entitled The Pathway-Based Interferonopathies, In from Cold. My name is Aksentijevich and I work at the National Human Genome Research Institute, Bethesda, USA. So regards to everyone from Washington. Today we will have three wonderful and inspiring speakers. Dr. Y.Crow from the MRC Institute of Genetics and Molecular Medicine at the University of Edinburgh, relation of type 1 interferon, development of biomarkers to response to treatment with TNF inhibitors. Submit your questions during this session via the QA functions. Enjoy the presentations.
Good afternoon. My name is Y.Crow. I'm a clinical geneticist working between the University of Edinburgh and the Aksentijevich in Paris, and I'm going to talk to you today about some novel type 1 interferonopathies. This is my disclosure slide. And my talk is actually structured around 5 points: an overview of the type 1 interferonopathies, a brief discussion of mutations in STAT2, a more extensive discussion of mutations in COP-A, a brief touching on the issue of clinical non-penetrance, and then some aspects of treatment in the type 1 interferonopathies. So that's a little graph of when you search PubMed with the word interferonopathy. So it's clear that since 2011 there's been a steady increase in interest in this field, even if this is a rather crude index of such. And this was where we were in terms of the number of type 1 interferonopathies designated somewhat arbitrarily in 2016. And this is where we are now, I would say. And I'd just like to go through certain aspects or highlight certain points in this graph. The first is in this slide. First is the interest that's developed around the use of disorders of DNA associated with DNA damage and DNA repair. And I mentioned the work of Nelson Gekara's group in 2015. Pointing out the induction of interferon associated with mutations in ATM, the work of Nicolas Manel and his colleagues relating to Bloom, and the work of Dr. Gössel and Dr. Gul and colleagues in relation to mutations in Artemis and DCL, REC1, RE1C, and also in ATM. So there's clearly a story that's developing around DNA damage, DNA repair, and the release of bits of nucleic acid that can stimulate the immune system. I'd also like to briefly point out some of these molecules that are associated with the lysosome, and I think that's interesting and maybe relates to, there are different mechanisms there. I'll also just point out here the possibility of the mitochondria representing a source of nucleic acids, which can represent an immunogenic substrate. The mystery that remains, but hopefully perhaps someone during the course of this conference will reveal in regards of the mutations in multiple components of the immunoproteasome or the proteasome, and the very strong induction that one sees in interferon signaling. And then I'm going to talk, as I mentioned earlier, about COP-A, touch on some of these or these molecules that are involved in post-interferon receptor signaling. And then I just highlight here, and I won't talk any further about these three molecules, ADA2, SEMD9L, and BAF, and whether or not they represent interferonopathies as one might define those. So another way of looking at all of this is to rearrange the picture. and think about ligand. So this is an issue about the production of ligand, and one might imagine one might suggest that in some cases some of these mutations are associated with the production of too much ligand or not enough or inadequate disposal of ligand, and that ligand can be generated from the nucleus or the mitochondria. Secondly, that one might produce aberrant ligand, and I think perhaps there's only one. I'm going to limit this particular point perhaps to a single example, which is mutations in ADAR1, where we don't adequately mark or edit double-stranded RNA species as self, and so they're misrepresented as non-self. And then there are mutations in molecules that are involved in involved in signal transduction, and the mutations result in abnormal signal transduction. And we all know about constitutive activation of STING. I'm going to talk briefly about mutations in COP-A, but there are also mutations in RIG-I and MDA5, which cause an alteration of the threshold at which these receptors respond to self-nucleic acid. And then finally, again, this idea that when you it's all good and well inducing an interferon response, but you also need to switch that off at the appropriate time. So I put this up. I'm not going to steal Dusan's thunder, but I just mentioned this lovely work from different people, from different groups, relating to mutations in USP18 and ISG15 as part of a negative control of interferon signaling. But I also want to draw attention at this point to STAT2. So STAT2 we recognize as a molecule that's involved in facilitating the interferon response post-interferon receptor. So in 2016 in my lab, when we put a patient from Tracy Briggs on our panel, we found these variants in STAT2, and I have to say I wasn't particularly interested in those variants as a cause of an interferonopathy phenotype, but it just goes to show that you need to keep up with the literature, because there was this lovely paper published in 2017 showing that actually STAT2 has as well as its day job, so to speak, it has a second function, and that function relates to the negative feedback cycle, and through an interaction with USP18. And then with the interferon receptor per se. So two wonderful papers from Chris Duncan, Tracy Briggs, and Sophie Hamilton, and Dusan, and I'm sure he'll talk about these further, showing that STAT2 has this very interesting function, and we can summarize that very briefly by saying that you've got an unstimulated state, and these molecules are resting. Then when you have full stimulation by interferon, we get the phosphorylation of STAT1 to STAT2, the interaction of STAT1, STAT2 and IRF9, and the subsequent induction of a broad transcriptional repertoire of interferon-stimulated genes. But I think what the mutations in USP18 and STAT2 tell us, and ISG15 also in that regard, is that there is a degree of tonic signaling, and the stimulus for such signaling, I think, remains something of an enigma, but it's clearly interesting, and why you do that is interesting. And essentially what happens with the STAT2 mutation is that the interaction, the stabilization and allowing of USP18, the interaction with STAT2 and the stabilization of USP18 and its interaction with the IFNAR2 component of the IFNAR is broken, and so you get a failure of that negative feedback. Okay, so in the second disorder I want to talk about in a little bit more detail is COP-A. And this is a very interesting disease that was described in 2015 by Tony Shum and Jordan Orange and colleagues. And essentially, there are three parts to the phenotype. The major, perhaps most devastating component is the lung disease. But there is also a joint phenotype, and there can be a kidney phenotype. Now, I don't think it's breaching any confidentiality now at this point to say that actually I reviewed this paper when it was first submitted to the New England Journal and before it was published in Nature Genetics. And I wrote this at the time. I wondered if this was really autoimmunity, what I really wondered was if this could be an autoinflammatory disease with something like Aicardi-Goutieres syndrome. This was actually just before Sting had been published. So I suggested to the authors they might want to consider an expression array looking for a potential type 1 interferon signal. So having reviewed that paper, I started looking out for copper patients, and this was a child. This was the first patient that we were able to ascertain. So this was an 11-year-old girl at the time who presented at the age of 2.5 years with joint pain and swelling and positive anti-nuclear antibodies. And then she developed some lung involvement with cough and clubbing, restricting her activities. And she had clear interstitial lung disease by the age of 6 years. And what she has is a maternally inherited mutation in exon 9 of COP-A. And I want to draw your attention, and I'll come back to this in a few minutes, that her mother had exactly the same mutation and was completely clinically asymptomatic. So you can see, you might just be able to read the figures here. So the first time we sampled this girl was in April 2016, when she was 9 years of age at that time. And we've been able to sample her over time since then, between the age of 9 years and 13 years. And it doesn't matter when we take a blood sample from this girl, and here's a 6-panel ISG, here's a 24-panel ISG. You can see this dramatic upregulation of type 1 interferon signaling in this child at every point that we test her blood. So here's another case. This is, I think, what people would consider the more classic copper case, but I don't think at this time we can really sort of be too dogmatic about what's classic and what's atypical. So this is a girl with disease manifesting initially at 2 years with life-threatening recurrent alveolar hemorrhage, and she can drop her hemoglobin very dramatically and bleed into her lungs. Infiltrating lung lesions on a chest CT, and she had a lung biopsy. And she, as far as I recall, doesn't have any joint problems, but she was also identified to have a heterozygous bona fide mutation in COP-A. And again, it was inherited from her mother, who was completely asymptomatic. And once more, you can see that it really doesn't matter when we take a blood sample from her. She has this very marked upregulation of type 1 interferon signaling. So here's a third clinical scenario. There's a family this time where all three individuals in 3 generations are symptomatic. So the girl presents at age 3 months with lung disease, which has got worse over time. The mother was diagnosed with rheumatoid factor positive polyarticular JRA in early childhood, and then she developed lung disease thereafter, diagnosed with ILD, and the grandmother was diagnosed with the similar type of joint disease age 16 years, and then she developed a lung disease, and eventually she died despite having a single lung transplantation. And I have to say, I thought these people had would have a copper mutation, but they don't. They have a STING mutation, so this is a classic Val155 MET mutation. And my point really here is just to say that I think that whenever you think about copper, you should think about STING and vice versa. So we can briefly summarize some of this, and and point out that you get lung disease in both disorders, and in copper it's more likely probably to be alveolar hemorrhage, but you can get the similar in, there's at least one case of STING SAVI described with that phenotype, but you get lung disease in both situations. Kidney disease so far not been a major feature of SAVI. And skin disease does not seem to be a feature of copper, but not everybody with STING mutations, gain of function mutations, has skin disease. And then joint disease is more common in COP-A. When you look at the biopsy specimens, there are some similarities between the two disorders, at least in certain cases. And I just point out, I keep putting up this slide, but I know I've added to it now. So these are hands that we've seen in people with mutations in TNase 2, in MDA5, in STING, and TREX1, and now in COP-A. And I put this up simply because I think what it shows is that there's this phenotypic overlap, which I would suggest is driven by the common pathological finding of enhanced type 1 interferon signaling in these discrete and disparate genotypes. So all of the mutations in COP-A fall within the WD40 domain, which is, so there's a very tight crux of mutations, and it's on the outside of the protein. It doesn't destabilize the protein, so it's there's no instability compared to mutations in genome in copper variants in COP-A found on genome AD. And the protein is present in these individuals, but effectively what happens is that this part of the molecule is involved in vesicular transport of cargo, of protein cargo. And what we think is going on is that what you've got STING here in the ER. STING traffics to the ERGIC, where it's activated. This involves COP2, but what our data suggests, and data of other people now, I'll mention this now, suggests that COP-A, as part of the COP1 complex, is involved in a retrograde transport of STING from the ERGIC to the ER. And that actually it means that there's a very low steady state of signaling. Obviously, when there's activation, the role of COP-A is overridden, but in the case of mutations in COP-A, it's as if the system is activated all the time. Now, the interaction between COP-A and its cargo is through something called a dilysine motif, and STING doesn't have a dilysine motif. And there are lovely data from Tony Shum and his collaborators to say that interaction between STING and copper is actually mediated by a molecule called SURF4. So we published our paper recently back to back with this wonderful paper from Tony Shum's group, where there's a mouse model and lung fibroblasts, really lovely stuff. There's a paper on bioRxiv from Tomohiko Taguchi, and also from Seth Masters' lab. So great deal of excitement around COP-A mutations. It's definitely a type 1 interferonopathy. You can see this tremendous upregulation of ISGs, and because of that, people are starting to think about the use of JAK inhibition in these disorders. So I just want to point out something else about COP-A, which is that, interestingly enough, there's a very high level of non-penetrance, clinical non-penetrance, and it's because non-penetrant asymptomatic individuals are not necessarily tested in a family. It's difficult to be precise about the figure, but we would estimate that between 20 and 30% of mutation carriers do not show a phenotype. And what we found interesting is that when we test the affected individuals who are symptomatic, they have an interferon signature. When we test those individuals that are essentially asymptomatic, they don't have, or they have a very mild, a much lower interferon signature. And you can see this also when we measure interferon. This is a composite. This is the interferon score plotted for a group of individuals. These are symptomatic individuals and asymptomatic individuals. So that's interesting. And that's actually quite different from what we've seen in MDA5 mutations. Again, a dominant disorder, again, where we found that maybe 13, so a less frequent, there's less non-penetrance in the families we've ascertained to date, but still very significant. But in most of these patients, most of these individuals who are clinically asymptomatic, we see very significant upregulation of ISG. So there are differences between these two disorders. I'm just going to finish off by highlighting the fact that there's a lot of excitement, a lot of interest around the use of JAK inhibition in the context of the type 1 interferonopathies. But I just want to leave you with a sobering lesson. This is a child who had AGS due to biallelic mutations in RNase H2B. And his sibling was diagnosed in utero to have the same two mutations as his brother. And we saw this child in Paris who was unaffected at the age of 4 months. So because of that, the child was started on ruxolitinib. And we saw a reduction in the interferon indices, never to normal, but we did see a significant reduction over the period of treatment. But unfortunately, this child then became symptomatic at age 14 months. So effectively what that means is that after 10 months of treatment on ruxolitinib at a stage, beginning at the stage where this child was clinically asymptomatic and developmentally normal, he developed Aicardi-Gouttier syndrome, albeit at an older age than his brother. So he has classic AGS, but just presenting at the age of 14 months. So I believe that JAK inhibition has its place in AGS and these other disorders, but I don't think it's the miracle cure that we need. And at least in regards of the diseases where there's central nervous system involvement, we're still searching for molecules that adequately penetrate the central nervous system. So I'll finish off there and thank all the people on this slide, in particular in regards to the copper disease, copper mutations. I'll point out the work of Marie-Louise Fremont, Alice Lepelle, and Pepper Niklos. I'd like to thank people in my Edinburgh lab, Joe Marsh for doing the modelling that I put up, and Gillian Rice as a long-term collaborator for her work on interferon signalling. So thank you.
Good morning, everyone, and thank you for tuning in to listen to this virtual presentation. I understand that I'm going after Y.Crow, and those are big shoes to fill, but I'm honored nonetheless, and I'll do my best. So today I will tell you about the USP18 and IgG15 circuit in human disease and how we have approached in dissecting the pathophysiology behind patients who have mutations in this circuit. So I'll first start with the notion that type 1 interferons are very potent antiviral and inflammatory cytokines. So they protect us from viral infections, but if untamed, they can cause severe inflammation that can lead to death as well. Type 1 interferons have a legacy of over half a century of discovery that started in mid-50s with the first publication that followed by the second publication, a little more famed one by Isaac and Lindelman. And then through a series of events in the 70s and 80s, interferon was first purified towards the end of the 70s, and then in the early 80s, Ian Gresser in Paris defined that type 1 interferon is not just an antiviral agent, but it can be neurotoxic in a series of beautiful papers published in early 80s. Also in Paris, Aicardi and Gouttier defined a syndrome in children, which we now know is Aicardi-Gouttier syndrome, And at the same time, David Levy, Nancy Ryan, and Darnell discover how interferon signals and which promoters it binds and what are the transcription factors that are being used. Then in late 80s, Sandro Pellegrini and George Stark, they developed the famed cells that we still use. They're deficient in part of interferon signaling. And then Pierre Lebon connects for the first time type 1 interferon to Aicardi-Goutier syndrome in a few nice publications. Then in the 90s, there was a series of beautiful papers coming from very influential groups that define really how type 1 interferon signals, how type 1 interferon is being induced, all the molecular events that lead to it. And then in early 2000s, Jean-Laurent and Stephanie described the first human STAT1 deficiency. And then in 2006, Y.Crow describes the first type 1 interferonopathy patients with mutations in TREX1. And then many discoveries follow since, but I'll stop here. In short, type 1 interferon signals through the receptor that is a dimer or interferon receptor 1 and 2. And in the intracellular domain, there are kinases, JAK1 and TYK2 that are associated with it. The transcription factor IgF3, which consists of STAT1, STAT2, and RF9, gets phosphorylated and translocates to the nucleus, binds to the SRE sequence, and then induces hundreds of type 1 interferon stimulated genes, ISGs, how we call them. Some of them function to negatively regulate the very system so we don't get to the over-inflamed state. Type 1 interferons have this early signaling, which is what I described. And if there are defects in this early signaling, if there is no type 1 interferon receptor, if there is no STAT1, there's no STAT2, there is severe susceptibility to infections. Mostly viral infections, but in case of STAT1, because it's a little more promiscuous, it's much broader. However, in type 1 interferon signaling functions also to control itself to prevent overt inflammation. And for that, we need cooperativity of IgG15, USP18, and STAT2. And I'll tell you today about that. They displace JAK1. And if this is not functioning well, then there is severe autoinflammation happening. And it really showcases that protection against viral diseases and infections and protection from autoinflammation. Okay, so I'll tell you first about IgG15, then about USP18, then about STAT2, and then about JAK1. They're going to be small vignettes, each about 4 to 5 minutes, as to the journey that we've been on for past 10 years and how we have approached it. So type 1 interferonopathies is introduced by Yannick and term coined by Yannick. Mendelian disorders that are characterized by persistent type 1 interferon-mediated inflammation. We can clinically diagnose them by looking at persistent expression of interferon-stimulated genes, so-called ISG signature. Clinically, they present with severe skin inflammation, systemic inflammation, intracranial calcifications, severe neurological symptoms, and it can be death. And here is a very nice timeline of discoveries of monogenic disorders that lead to type 1 interferonopathy. And I will tell you about those one in red that I have been and my group has been involved in characterizing. Okay, so type IgG15 deficiency, here I'm showing you the latest 5 patients that we have discovered from 3 different continents and 5 different countries, or 4 different countries and 3 different continents. And unifying in all these patients is that if they receive a BCG vaccine, they develop disseminated BCGiosis. Then they have necrotizing skin lesions that are usually self-resolving, and all of them have basal ganglia calcifications that can be detected pretty early on in childhood, which we know is unusual. However, these basal ganglia calcifications, unlike in other type 1 interferonopathies, do not seem to to affect the neurocognitive behavior of these patients whatsoever. So it's very mild. Here is the CT scan of some of the patients with ISG15 deficiency with idiopathic basal ganglia calcifications of Farr's disease before we knew it was really type 1 interferonopathy. Earlier this year, we published these new 5 patients because in addition to this infectious phenotype of susceptibility to BCG, and in addition to their intracranial calcifications, they have necrotizing lesions, and they usually show in the pubic area and the neck area and in the early childhood. And we looked deep into it, and it seems to be that the keratinocytes are inflamed, that there is infiltration of myeloid cells in the tissue, and then also the inflammation of vascular tissue. In peripheral blood, all of these patients, as we reported before, have elevated type 1 interferon-stimulated genes. If we perform single-cell RNA sequencing, we can see that there is an elevated IgG expression in the absence of IgG15, and it's particularly evident in monocytes and dendritic cells. So here I'm just illustrating the signature of interferon-stimulated genes in classical monocytes and comparing healthy control to IgG15 deficient patients. Here I'm illustrating that usually these patients have a complete loss of protein expression and function. As you can see here in EBV-derived B cells from this patient, there's completely lack of expression of ISG15. However, it's very interesting genetics that we've discovered in a number of these patients. They do all follow recessive mode of inheritance. And in these 11 patients so far described, there's 8 different genetic lesions, and they are homozygous top, homozygous frame shift, compound heterozygous, missense mutations, and essential splice site mutations, which makes for a beautiful genetic study in these unfortunate patients. IgG15 is not just a free molecule, it's also ubiquitin-like molecule which can tag to proteins to form this acidulation pattern like ubiquitination does. So in absence of IgG15, there is no this smear that we can detect here. If we derive cells from these patients and inflame them with interferon and look at the resolution of type 1 interferon inflammation, you see how patients who don't have IgG15 cannot resolve this inflammation, here measured by MX1. However, if we introduce back the wild type allele, we can completely rescue this phenotype. As you can see here, unlike if we transduce IgG15 deficient cells with a control vector that expresses just RFP. How does this mechanistically function? When there is no IgG15, the stability of USP18, which is this master negative regulator that displaces JAK1 at type 1 interferon receptor site, is unstable. So in a way, complete loss of IgG15 leads to hypomorphic level of USP18 and its function. And in absence of it, it gets more proteasomally degraded. And we have noted that a few years back that mechanism in collaboration with Sandra Pellegrini. So we have also noticed that because these patients are largely doing well, they have this infectious episode, an inflammatory episode very early on in their childhood, they actually have increased resistance to viruses. And we have published that in 2016. Next is the USP18 deficiency, and that phenotype is much more stark than that of IgG15 deficiency. Initially, we reported 2 families with complete loss of USP18, and these patients presented also with necrotizing skin lesions. They were septic, they had ARDS, they were on ventilator, they presented vasculitis, hydrocephalus, and death, and they had basal ganglia calcifications. As you can see here in imaging of their brain, it's a very severe phenotype. And then here, looking at the Western blot, it's a complete loss of expression of USP18. If we looked at the relative amount of inflammation, you can see here, as measured by IFIT1, that compared to controls, they are unable to control type 1 interferon-mediated inflammation. If we look at the brain sections of these individuals post-mortem, we see that phospho-STAT1 is upregulated in microglia of these patients. In their cell lines, if we introduce back the wild-type USP18 allele, but not the control allele, we are completely able to rescue this type 1 interferon-mediated inflammation. I told you that ISG15 leads to these smears that are ISG-related proteins, where USP18, in addition to being at the receptor site displacing JAK1, is also an enzyme, and it helps in de-ISGulating, removing enzymatic ISG15 from proteins to which it was conjugated. So here you can see that this patient has hyper-ISGulation as well, and then when we introduce the wild-type allele back, that is, of course, reduced because ISG15 retains its normal function. We also saw the reversal of interferon signaling as measured by phosphostat1 and phosphostat2 in presence of wild-type allele and not of luciferase. And we published this in collaboration with Grazia Mancini at Erasmus University in Netherlands. 2 years later, we came across another patient who was admitted to PICU with septic shock-like features, and over the course of a few months later developed ARDS, or intraventricular hemorrhage, necrotizing cellulitis, which you can see here, and subacute infarction in the brain. At 7 months old and very, very heavy PICU monitoring, which really saved his life. He was whole exome sequenced, and homozygous mutation in a splice site in USP18 was identified that putatively led to deletion of exon 10. That we confirmed using Sanger sequencing that the mode of inheritance was autosomal recessive. And when we did the 3' race, we identified that truly there was deletion of exon 10 in the patient. This deletion led to normal protein level expression, but inability to control type 1 interferon. Because if we introduce the wild type back into the cells become hyper-responsive to interferon, but this delta exon 10 USP18 that this patient carried was unable to perform its function that would displace JAK1. Interestingly, and I'm not showing here, this patient had largely normal de-ISGulating activity, suggesting that the hyper-ISGulation in itself probably doesn't contribute hugely to the phenotype we have detected in this patient. Perhaps most importantly, this patient was put on JAK1 inhibition, and disease completely resolved within a few months, and the kid is now 3 years old and actually doing very well, even with cognition improving. And all of this was really done by Fahad in Saudi Arabia and Marta in my lab, and it was just a showcase of a beautiful transcontinental collaboration. Okay, so far I've shown you that USP18 deficiency is very stark, and it can lead to death through very, very strong induction of type 1 interferon response. Whereas IgG deficiency is much milder, it's compatible with life, usually self-resolving, and perhaps even protective against viral diseases. Okay, so I've told you first 2 vignettes about IgG15 and USP18, and now I'm going to tell you about the discovery of STAT2. So this patient presented with necrotizing skin lesions, basal ganglia calcifications, septic shock, ARDS, was put on ventilator, vasculitis, had hydrocephalus, so really mimicking USP18 deficiency, which led to death. So unfortunately, the patient wasn't diagnosed in time, but the discovery led to homozygous mutation in STAT2, which is very interesting, and I'll come to that in a second. This is the clinical picture that you can see of the patient's brain, the amount of skin inflammation, and how the lungs looked. We derived a cell line from this patient and inflamed it with type 1 interferon. And as you can see here, healthy control levels of IFIT1 are, about 500 relative units of IFIT1, whereas in the patient, it was significantly higher. So this became peculiar to us because canonically, STAT2 is a positive regulator of type 1 interferon signaling. So if there was a mutation to be had, and the ones that have been described in STAT2, a patient should have hypomorphic response to interferon, not hypermorphic, which was the case here and with clinical presentation of the patients. But then we thought about the late response interferon, and it was described before in 2017 that the STAT2 doesn't only positively regulate type 1 interferon, but also helps recruit USP18 once it's synthesized to the receptor to bring it. So in a way, it is a positive regulator initially in the early response, but in the later response, it may act as a negative regulator as well, as somebody who carries USP18 to exert its function. So we first wanted to test the requirement of homozygosity for this phenotype, and indeed homozygosity was required for this phenotype. Then we assessed the early responses, the phosphorylation, dephosphorylation, translocation to the nucleus, and it was completely normal, which is what it needed to be. And then the late response was indeed abnormal because this STAT2 mutant was unable to efficiently, unlike the wild type, carry USP18 to the IFNAR2. So indeed, that was the mechanism by which this mutation affected negative regulatory ability of STAT2 via recruitment of USP18 to the receptor. So that's how this patient presented with such strong autoinflammatory syndrome. Okay, the last story is that one JAK1 mutation, which is of course not only involved in type 1 interferon signaling, but many others. This patient presented with ILVEN, which is nevus that follows biological lines of development, and they'll become relevant in a second, with non-specific enteritis, colitis, esophagitis, with eosinophils in the lamina propria. Patient had membranous nephropathy, kidney transplant, recurrence of membranous nephropathy, skin disease, and kidney rejection. We identified the mutation de novo in JAK1. JAK1 doesn't just signal through type 1 interferons, but through a family of over 25 cytokines. So what did we do? We first used cells that were made in the 80s by Sandra and George Stark that don't have JAK1. We transduced luciferase, and when given interferon, obviously these cells cannot signal. When we transduce back wild-type JAK1, we can restore the signaling of STAT1. But when we transduce the mutant, you can see that even in the absence of type 1 interferon, there is phosphostat1, suggesting that this is potentially gain of function. So how is this gain of function? JAK1 is known to cis-regulate, and this pseudokinase domain regulates the kinase domain. And indeed, here we can see that there is more of phospho-JAK1, so this made sense. But we've also wanted to test the other hypothesis, that the pseudokinase domain of one JAK was trans-activating, trans-regulating the partnering JAK. So to do that, what we did is we created a mutant that's kinase inactive in JAK1. And here are the results. So luciferase cells that are JAK1 deficient cannot signal. If we introduce the wild type allele, we can restore the signaling. If we introduce the patient allele, there is signaling in absence of any interferon, here interferon gamma. If we create this kinase mutant, there is very low amounts of JAK2 signaling, because JAK1 is incapable of doing it, but it's enough for a scaffold. So it's extremely hypomorphic and all coming through JAK2. And then if on this kinase mutant we introduce the patient mutation, we still see gain of function, suggesting that it was also regulating the partnering JAK. Okay, we profiled the immune system of this patient and saw that this level of inflammation was present at the basal state in absence of any cytokines, which we didn't detect. We measured phosphostat-1 and a bunch of other phosphostats. But the point I want to make here that phosphorylation of STAT1 downstream of IL-2 should not happen. And in this patient, we're detecting it. So this is not just augmentation of already existing pathways, but it's neomorphic. There is new pathways that are being activated by this gain of function. We profiled phosphostat-3, 5, and 6, as you can see as well. And last part of this patient is that this patient turned out to be mosaic, and we project that this mutation happened prior to gastrulation because it's seated in all three germ layers. We cloned both wild type and the mutant cells from the patient and replicated the phenotype I showed you before. So the wild type cells from the patient, there was no basal signaling, and those that had the mutation had basal activation. We performed single-cell RNA sequencing and identified that NK cells are more resistant to this mutation. And perhaps most interestingly, I'll skip this slide, we saw that JAK1 is expressed in biased monoallelic fashion. So we expected wild-type cells just to have the wild-type allele, and we expected the heterozygous cell to have the wild-type and the mutated allele. But again, here there was mostly the mutated allele, which is not what should happen this is how a gene should look if it's a heterozygous. This happens to be true not just for this mutation, but apparently for JAK1 in a silent SNP that we measured here, that there is this monoallelic bias happening, unlike in the control gene. We tested JAK1 inhibitors in vitro, ex vivo, and then placed the patient on an FDA JAK inhibitor. And within 2 months post-initiation of tafasitamab, there is marked reduction in phosphostats across many cell types, or reduction in ISGs, reduction in C-reactive protein and SED rate, resolution of clinical disease. You can see here pre and post-treatment, and you can read it in Immunity that just came out in August of this year. Unfortunately, this patient was exposed to COVID and died of COVID earlier this year because, she had transplant, she was on tacrolimus, and it broke our heart. But for 2 years, her quality of life was immense. Here are the conclusions that I told you about already, and here's the acknowledgement slides. I don't have any disclosures, and here are Connor and Marta, who really led most of the work that I presented here. Thank you for attention, and I am looking forward to live chat in a second.
Thank you very much for the invitation to give you an update on the preclinical studies for gene therapy in a condition called deficiency of adenosine deaminase type 2. We've been working on this project for a few years now, so I'm going to be presenting some preliminary results of the experiments we have undertaken. I'm Despina Eleftheriou, and I'm a professor of pediatric rheumatology based at UCL Great Ormond Street Hospital Institute of Child Health in London, UK. And I have no relevant disclosures to this work. So the disease deficiency of adenosine deaminase type 2 was described in 2014 with 2 very important publications in New England Journal of Medicine. They were describing a genetic inherited disease that looked like polyarteritis nodosa, and the patients were presenting with early onset stroke. What we now know is that the disease is caused by biallelic loss of function mutations in ADA2, and the gene ADA2 encodes for the ADA2 protein that has a partial homology to ADA1. You may be familiar with ADA1 because genetic lack of ADA1 causes severe combined immunodeficiency, and this is one of the first conditions that were really treated with gene therapy. Both ADA1 and ADA2 act on the same pathway of converting adenosine to adenosine. The disease we know is associated with reduced ADA2 protein concentration in most patients, and also this results in a reduction in enzymatic activity in blood. There's no mouse orthologue, but the zebrafish knockdown present with intracranial hemorrhage and neutropenia, resembling really the symptoms we see in humans. So what do we think is the pathogenesis of the autoinflammation and vasculitis associated with this disease? A number of in vitro and ex vivo studies have shown that there's aberrant monocyte differentiation, favoring a polarization towards the pro-inflammatory M1 macrophages. There's also upregulation of neutrophil-related genes, including myeloperoxidase, and an induction of interferon gene expression. We also know that ADA2 is a growth factor for the endothelium, so there's a pro-inflammatory effect on the endothelium, probably indirect, mediated by monocytes and neutrophils. So this schematic really represents this imbalance between M1 and M2 macrophages as just referred to. So there's increased death and apoptosis and aberrant differentiation of M2 macrophages. So we get lots of numbers of M1 macrophages producing a number of cytokines such as interleukin-1, beta, interleukin-6, and importantly TNF-alpha that disrupts the endothelial cells, as you can see in this picture here, and causes vasculitis. What's less understood, well understood, is the pathogenesis of immunodeficiency. Some papers suggest there's reduced memory B cells, reduced terminally differentiated B cells and plasma cells, and an increased mortality of B cells. There's inconclusive studies about the T cell dysfunction, but certainly some studies are suggesting the T cell function is affected too. So importantly, therefore, both B cells and T cells may be affected, and this is important for the gene therapy strategy we have taken. The main clinical features of the disease, we see a number of different phenotypes. So there's the autoinflammation vasculitis phenotype, with patients presenting with recurrent fevers, levodoracemosa, and polyarteritis nodosa, neurological phenotype with lacuna ischemic strokes, intracranial hemorrhage, and peripheral neuropathy, and the more difficult to treat immunodeficiency and bone marrow failure phenotypes. It's important to also highlight there's variable penetrance, so there's some patients who may be pre-symptomatic and may only develop symptoms in adult life. So here are some pictures of the symptoms we see in this condition. So this young boy was age 14, and he was presenting with cutaneous vasculitic lesions, as you can see here, and digital ischemia. He also had a foot drop, and on sural nerve biopsy, shown here, he had frank necrotizing vasculitis, suggesting polyarteritis nodosa. He was subsequently found to have biallelic loss of function mutations in ADA2. Another patient presenting with an angiogram that we used in several textbooks to talk about polyarteritis nodosa, but actually was secondary to ADA2, and was also presenting with this hemorrhage, intracranial hemorrhagic stroke. The levodoracemosa we talked about, you can see in this picture, this patient was also presenting with an unhealed cutaneous ulcer. And this last patient here was actually presenting with sclerodermatous skin disease, and that's the working diagnosis, but then was found to have ADA2. So what are the treatments available currently? And there's no formal trials. This is a rare disease. Corticosteroids may have a role, but other treatments that we use for autoinflammation, such as colchicine and anakinra, are not effective, and neither are conventional treatments for polyarteritis nodosa. What we know, though, is TNF-alpha is effective, given the mechanisms I described affecting macrophage differentiation and secretion of cytokines, for treating the autoinflammation and vasculitis associated with the disease, but probably not as good for marrow failure, and there's papers now supporting that, and possibly not for immunodeficiency. We also have seen some patients lose efficacy over time. Very few, but that is still happening. So allogeneic stem cell transplantation has been used in some cases and is curative. Of course, there's important caveats around undertaking a stem cell transplantation. So why consider gene therapy? Well, the fact that this is a recessive monogenic disease leads us to hypothesize that gene addition to restore ADA2 protein and enzyme activity may treat and cure the disease. The fact that stem cell transplantation has been successful suggests that targeting CD34 positive hematopoietic stem cells for gene therapy could be an approach we could take. Whilst there is some endothelial expression, this is less important given that ADA2 is an extracellular enzyme, and protocols already exist for successful ex vivo viral delivery to stem cells for other diseases, for instance, ADA1 SCID. So on the background of this, we hypothesized that using gene transfer strategies, it is possible to correct the defects associated with ADA2. Lots of the data I'm showing you here are unpublished, so please do not copy or distribute. And I'd like to thank Ying Hong and Marina Casimir for producing this data. So our first objective was to actually create a knockout cell line that we could use in the lab to replicate the immunophenotype we were seeing in cells derived from patients, and also see if with gene transfer strategies we could correct these defects. So we used CRISPR-Cas9 and we created a cell line successfully with complete ADA2 knockout. You can see the clone analysis here. You can see the protein expression, absent protein compared to actin here, and also reduction, complete absence of ADA2 enzyme activity. We, of course, recognize that creating cell lines that have specific mutations we're seeing in patients are also important. So we now have, with CRISPR-Cas9 technology, 2 cell lines using the more common mutations we see in patients in THP1 cells, and we're working with the cells too. I'm aware there's a lot of different studies of looking at the pro-inflammatory phenotype we see in monocytes and macrophages in these patients. This is just one approach and the approach we've taken. So THP1 cells were treated with PMA and then monocytes, macrophages derived that then were polarized into M1 and M2 lineage using a cocktail of cytokines and chemokines. And then we looked at gene expression of a number of pro-inflammatory cytokines and molecules. And you can see that for TNF-alpha, that the wild type cells had reduced expression compared to an increase in TNF-alpha we were seeing in the ADA2 knockout THP1 cells. The same for interleukin-1 beta, STAT1, and CXCL10. We also looked using the same model at secretion of TNF-alpha, and you can see here the knockout cells have a significant release in the culture supernatant of TNF-alpha compared to the wild-type cells. We were also interested in the anti-inflammatory phenotype of the cells, the skewing of M2, M1 ratio that we are seeing in patients. And you can see in the knockout THP1 cell line, we're getting a reversal of this ratio compared to what we see in wild-type cells. And this is exactly what we were seeing in patients in monocyte-derived macrophages obtained directly from patients with a number of different mutations. So increase in gene expression for TNF-alpha, increase in CXCL10, increase in TNF-alpha release in the supernatant, and also reversal of this ratio of M2 versus M1. So a good cell line model to study the disease, given that it replicates the immunophenotype of what we're seeing in patients. So then we wanted to develop lentiviral vectors and explore with appropriate promoters whether these are effective with gene transfer into correcting these defects. So we collaborated and are still collaborating with Professor Thrasher and Bobby Gaspar's group. We used the exact same backbone of vectors that have been used in SCID clinical trials, tagged to express ADA2 and GFB, as you can see here in this slide. So then we use this lentiviral vectors to look at both cells derived from patients and THP1 cells and explored a number of different readouts. So very firstly, we looked at the expression of the protein, and as you can see here, transduction of either wild type or knockout cell line cells was restoring the actual protein expression, and there's nothing happening when you use GFB, and the wild type cells were not affected. So good restoration of protein expression with transduction of these cells. We also looked at enzymatic activity, and you can see in the schema that enzyme activity was recovered with transduction of the ADA2 knockout THP1 cells, as shown in this graph here. We also looked at TNF-alpha release. Remember, all these cells are very pro-inflammatory and release a significant amount of TNF-alpha. So you can see that the corrected cells, this was suppressed compared to the uncorrected cells, almost to levels that we were seeing in wild-type cells. We then moved on to look at patients. We use flow cytometry to assess protein expression, and we use monocytes derived from a number of different patients with different mutations, converted them into macrophages, and then looked at ADA2 expression. And you can see that they transduced corrected cells. There was recovery of ADA2 protein expression compared to uncorrected cells, almost levels what we're seeing in healthy donor cells. We then looked at the number of other immunophenotype readouts, so TNF-alpha expression. Again, you see in corrected cells this is suppressed, and these are the individual really experiments and how this changed for each experiment over time. Similar trend in other cytokines, perhaps not in interleukin-1 beta, but no surprise there because we do know from clinical experience that anakinra is not really effective for these patients. We looked at M2 macrophage phenotype as well, initially starting with the apoptotic assay. So you can see the actual transduction of the cells was leading to reduction in apoptosis of M2 macrophages. And then when we took the supernatant from these M2 macrophages and incubated with endothelial cells, looking at endothelial activation expressed as CD62 on flow cytometry, we could see that this was recovered in the corrected cells and there was no endothelial activation. We're now moving on and have started studies with CD34 hematopoietic stem cells, which will be the target of the actual clinical gene therapy studies. We're using healthy control cells, but also have some cells from patients. And the idea is to look at transduced and untransduced cells and answer two important questions relating to ADA2 expression and enzyme activity and immunophenotype, as in the experiments I've just shown you in cell lines and patient cells, but also whether these cells maintain the stem cell properties and look at the viral copy number. So these are preliminary experiments looking at healthy cells, and you can see transduction increases the protein expression using a different MOIs in here. And very importantly, looking at cell proliferation assays and also CFU numbers in different lineage cells, these are maintained. So the cells following transduction have an increase in protein expression for ADA2, but also importantly, maintain the stem cell properties. The next steps for us are working on in vivo studies, looking at NSG mice and some engraftment studies. We will take the approach of looking at various organs in terms of histopathology and biodistribution, but also then hematopoietic organs, peripheral blood, marrow, spleen, and thymus, to look at engraftment, biodistribution, histopathology, and importantly, ADA2 expression, enzyme activity, and immunophenotyping of macrophages. So I hope in summary I've shown you that we have a very successful in vitro model to recapitulate the immunophenotype of ADA2. We have some lentiviral vectors encoding ADA2 that we have shown in both the knockout cell line model, and we also have data on the specific inserted mutation cell lines, but I haven't shown these today, as well as monocyte-derived macrophages from ADA2 patients that can correct the protein expression and enzyme activity. We can rescue the immunophenotype in these preliminary experiments, and I'm sure there's a lot of other readouts that may come to mind and may be useful for us to do, but also reduce the inflammatory interaction with endothelial cells. It's important that we now complete our work on stem cells, which will be the target cell for gene therapy. Before then, we work with all of you to identify who could be the patients we can target and move on to some clinical studies. I'd like to thank my collaborators, Paul Brogan, Ying Hong, who generated this data, and the number of other collaborators we have at the Royal Free, as well as the NIH, Harry Malek, and also a number of funders who have supported this work. Thank you for listening.
Good morning to everyone.
Hello.
Hello.
I guess now we are on live streaming. I'm not sure what's happening. Should I start with the questions?
I think so, Ivana. Yeah.
Yes. Well, first I would like to thank you, our wonderful speakers, for their inspiring lectures. And I see that we already got a few questions for all of you. So I will just read them in the order how they were submitted. So the first question is about the ISG15 deficiency, and the question is for Dusan. What is the mechanism of Mendelian susceptibility to mycobacterial disease in patients with ISG15 deficiency?
I love this question. I can answer it partly correctly and partly we're working on it. So what I haven't told you during the talk is that ISG15 also gets secreted, and as a secreted molecule, so there is intracellular activity of stabilizing US-18, the ISGlates, so conjugates to other proteins, and that's intracellular functions. It also gets secreted, and as a secreted moiety, can stimulate lymphocytes, especially in combination with IL-12 to produce interferon gamma. So if we test whole blood from ISG15 deficient patients, we always see a slightly hypomorphic production of interferon gamma, and that's where we think the MSMD is coming from. Having said that, we are also actively working on a second mechanism which we think contributes to MSMD, and is also seen because, what is it, about 15% of STAT1 gain of function patients also have MSMD. So there is something about type 1 and type 2 interferon signaling competition which attenuates one or boosts the other one based on timing, and that we don't quite understand yet.
Okay, that's interesting. For sure. The second question is a long one, and again for Dusan. So how is associated type 1 interferon upregulation with autoinflammatory skin lesions? Or I suppose the question is, what is the mechanism of skin inflammation in patients with upregulated type 1 interferon production? There is any evidence about autoinflammatory skin lesions in patients under recombinant alpha interferon therapy? What have you seen in the single-cell transcriptomics behavior of other non-type 1 interferonopathy or interferon inflammatory pathways in those patients? What are your thoughts about the therapeutic potential of targeting USB18 STAT2 pathway in those patients with interferonopathies and increased susceptibility to infections?
Okay, so simple questions. I would love to know how interferon affects the skin. I don't think we know. We are only starting to characterize that. At least in ISG15 deficiency, we know that keratinocytes are inflamed, we know that endothelial cells are inflamed, and we know that there are infiltrate of CD68 positive macrophages. I think the skin inflammation is typical for all type 1 interferonopathies and beyond. And I think mostly we don't know what's going on, but we're, I guess, working on it. What do I know about individuals who are receiving recombinant type 1 interferon? Whether do they develop any skin lesions? I'm not aware that's the case. I guess the best example would be interferon beta treatment in individuals with MS, and maybe there is some, but I'm unsure. And in single-cell transcriptomics, when we looked and saw this clear upregulation of ISGs in myeloid cells, well, mostly monocytes and dendritic cells, we didn't see any other notable dysregulation that was obvious, right? And sometimes you have to ask the right question to get the right answer. So it's not staring at us, but it doesn't mean it's not there. But for now, we don't see it. And the last thing is, yes, I do think for the USB18, JAK1 inhibition was really life-saving for this child. For the STAT2 gain of function, we didn't diagnose it in time. So in that sense, I think the therapeutic benefit heavily outweighs the potential immunosuppression that this can cause.
Okay, thank you.
I'll just add, there is a literature about the use of interferon and chilblain-like lesions, so that's been described. What strikes me, Dusan, is the skin lesions that you're seeing in it wasn't the ISG15. Those sort of necrotic lesions in the groin area. That's very. I've never seen that in AGS.
Right. There is something slightly different. I agree. I absolutely agree. There is something there, and we can't quite point our finger to it.
Okay, thank you. The next question is, were all necrotizing fasciitis skin inflammation patients in your series without inciting pathogens like in familiar autoinflammatory fasciitis by NF-κB JAK1 mutations, or were you able to isolate typical bacterial pathogens?
So for most of them, we couldn't isolate anything. For those that we isolated, they were just common skin commensals. So, whether it is the very commensals that are, introducing some sort of a tonic interferon signaling that goes out of whack, it's very possible. But it's not a particular pathogen that we are fairly certain of, or commensal.
Okay, thank you. Next question is for Yannick. So have serum or CSF levels of ruxolitinib been measured in that kit before and during the flare?
So this is work that Benedict Nevin has done in particular. What we do, all I can talk about is ruxolitinib, not baricitinib or any of the other JAK inhibitors, but essentially the levels of ruxolitinib in the CSF are about 10% of those levels. When we measure simultaneously at the same time, the levels in the CSF are about 10% in the blood. So there is penetration, at least with that drug, but the amount of drug in the central in the CSF is lower than compared to in the periphery. And I think maybe increasing the dose could increase the amount of penetration. I think that issue is a very important one. I mean, clearly, peripherally, at least in relation to certain aspects of these diseases, the JAK inhibitors have a positive effect. I think that is clear. We're barking up the right biological tree, as I sometimes say. Although, I would also say that these are not just specific to JAK1 inhibition, and we can't say for definite that it's due to that mechanism. But I think one issue is about penetration, and I don't know if you can get around that by increasing the doses so much. I just don't know. That's one.
So maybe a vignette, Yannick, is from the USP18 deficient patient from Saudi Arabia. For that one, Fahad and Abdullah first started on 5 mg of Rux, and it was completely unresponsive. And then they upped it to 10 and complete resolution. So there is something in those in our hands.
Yeah, no, that's right. You used double the dose that we were using. Yeah.
Okay. The next question is for the SPINA. Do we know of any possible DADA2 deficiency different from classical ADA2 biallelic mutations?
Okay, so Ivona, you may want to comment as well. I hope I'm understanding the question correctly. I think you're asking about genotype, phenotype, and functional correlates. And there's an important publication we all contributed to, published earlier this year by Pui Lee, who looked at actually a number of different patients from around the world, around 150 different patients. And the conclusion from that was that actual missense mutations in ADA2 that still maintain some residual enzyme activity were mostly associated with the vasculitis autoinflammation phenotype, whilst nonsense, missense, insertions, deletions that were actually leading to complete loss of function of ADA2 and enzymatic activity were really mostly correlating with a phenotype of severe hematological manifestations, the patients that were really struggling to treat.
Yeah, that's correct. So I thought that was the question. I didn't understand this question in any other way. So there is a lot of phenotypic variability in patients with ADA2, like is the case with many other human diseases, but we can at least make some prediction based on the nature of mutations, what's going to be the phenotype, developing phenotype in these patients. So the next question is again for the SPINA. What promoter did you use for ADA2 expression? Are you worried about possible overexpression of ADA2 in CD34 positive cells?
So we're using an EFS promoter, so the exact same promoter that's been used in the SCID ADA1 clinical trials, just I guess because in our institute we have people who have been pioneers in clinical trials in ADA1 SCID. So we're hoping there's not going to be any problems, but I guess we need some more ex vivo and in vivo studies to 100% confirm that's the case.
Okay, thank you. So, Despina, I have a question for you. So, how are you going to select for the patients that should be, having gene therapy based on what? And also another question is there any, pardon or excuse for my lack of knowledge for, gene therapy protocols, but is there any age limit, that may prevent some patients, for example, from undergoing this procedure?
So we just started having the conversations about the actual clinical part of the study and trying to address some of the important questions you're actually raising now. I think what we're seeing clinically is the patients with severe immunodeficiency and severe hematological phenotype that tend to be refractory to TNF-alpha, and perhaps other ones we should be targeting for gene therapy. The caveat and the difficulty here is that if you have severe aplastic anemia, for example, can we convince ourselves that we will be still able to mobilize CD34 cells peripherally, and that's an important clinical conundrum we still have and we will need to address in clinical trials. I think in terms, the second part of your question was about protocols.
No, my second question, is there any age limit, when selecting patients for these procedures?
So from other studies in the field for primary immunodeficiencies, the sooner the better, but still patients in young adulthood are getting transplanted and still are getting approached for gene therapy. So I don't think age will be an important limitation, but I guess what we don't want with all these processes, hematopoietic stem cell transplantation, gene therapy, we don't want patients going into this with severe morbidity acquired along the line. So, patients who have got significant sequelae of their disease are not the best to undergo such a process. So perhaps targeting patients earlier on might be best from that perspective.
Mm-hmm. Okay, thank you. Let me see whether we have more questions. I don't see them. I have a couple of questions. So, Yannick, you mentioned something about this SIRF4 protein, right?
Yeah.
That serves as a adapter protein and interacts with COPA-A to basically shuffle STING from Golgi to ER, right?
Yeah.
Okay, so is there anything known about what would be the effect of knockdown of SIRF4 protein in some cells, whether you do it in, THP1 or HEC293 cells, and whether, that would lead to upregulation of the interferon?
Yeah, I think it would be the same effect, actually. We I don't know if you know, there's a molecule called STIM1, which it's got a dilysine motif and is said to bind to STING. And we thought that was the candidate, and we tried to, but when we knocked that down, nothing happened. But I think the SIRF4, I'm guessing if you had loss of function mutations in SIRF4, you get the same phenotype, actually, yeah.
So would you expect to find the biallelic or dominant mutation in SIRF4? Because this is an adapter. Yes, I would expect to find biallelic, or recessively inherited mutations, although this phenotype may not be viable. I don't know.
Yeah, sure. I really don't know. The copper is a dominant disease. I have to plead ignorance. I haven't looked to see if there's a SIRF4 mouse, for example, or whatever. But I think, it's very interesting. The Schum paper is really a delightful paper, I have to say, and it's very interesting. So they have a biallelic copper. The biallelic copper knockout mouse just doesn't make it, is non-viable, but they don't say much about it, but they salvage the mouse when the copper mouse when they take out STING. So it's very convincing, actually, and they had access to patient lung fibroblasts, which was also rather lovely, I thought, to be able to look for a signature in that cell type. So I don't know much about copper, SIRF4, but it's an interesting point. Yeah.
So when you get patient referred with possible interferonopathy, so what is your workup of this patient? What do you do first? Do you, do a CT scan to look for basal ganglia classification? Do you do interferon signature, or you just test these patients for mutations in,
Well, I think we've learned enough now about. There are certain phenotypes which I think evoke the story, and fairly easy to diagnose now, and maybe you just go to gene sequencing. But I think what we've tried to do, I mean, the kind of USP, is that right, of the lab has been to sort of be indifferent, if you will, to the phenotype, and look for the signature.
Mm-hmm.
And if you find the signature once, you look again for the signature, and then if you find it 3 or 4 times, you know that you're onto something, and then do the sequencing.
Mm-hmm.
And even if that's a gene that's already been described, like copper, described a long time ago, but several years ago, but Stefano's paper, and then the work that we've done and other people have done now, that only by looking at the transcriptome do you get the story. I mean, it's the same with the PSMB8, isn't it? I mean, Garg published the gene in the American Journal, but it was only later on that Raffaella and her people look for the signature. And I think in a way, very generally, I would say that we kind of neglect, transcriptomics as one way of trying to sort out what's going on. And luckily, but also perhaps slightly dangerously, the interferon signature is a very big, bright signature, as we've big bright signal as we've discussed, and maybe you get sort of taken in by the brightness and you forget about other things that are going on.
Yes, I agree with you. We got another question, and that is, did anybody already diagnose ADA2 deficiency without any ADA2 mutations? Spina, I guess this is the question for you and for me.
Yes. So I'm sure you'll make a comment as well. I mean, my understanding and approach to this is that obviously if there's absent ADA2 enzyme activity and you're looking at the patient with a very typical phenotype, then you need to look at the genetic data harder. And there's papers out there, I guess, acknowledging that deletions might not have been picked up initially, some cryptic mutations, etc. So I think the first approach would be look at the genetic data again. However, I guess there may still be the case scenario that we're seeing a resembling phenotype and still absence of ADA2 enzyme activity for diseases that may still mimic what we think is deficiency of adenosine deaminase type 2, and that opens up, I guess, an approach for further looking at other candidates. But I would first look at the genetic data carefully, is my approach.
I agree with you. Thank you. Let me see whether there is another question. I don't see any new questions. So Dusan, you mentioned something about that ISG15 mutations may confer resistance to viral infection. And so are these pathogenic variants that are found in patients with ISG15 deficiency novel, or they are kind of, rare in the general population? So what I'm thinking about is whether there is a possibility that maybe these mutations could have been positively selected throughout human, evolution for the reason that they confer resistance to viral infections.
Right. I've been thinking about that a lot. So genetically, we have no evidence of positive selection whatsoever. They're exceedingly rare, and they're all biallelic and recessive mode of inheritance that lead to essentially complete loss of function in all but one case. In that one case, it's severely hypomorphic. We estimate about only 5% of the function of the gene, right? So in that sense, we don't think there is an evolutionary advantage to it, and the answer might actually be the mycobacterial disease, right? Because if they're susceptible to mycobacterial disease, then there was big purging historically, in the context of TB and perhaps here even environmental mycobacteria. But having said that, unless the patient presents to a clinic with a phenotype, we don't look for what's going on there. And nobody goes to the doctor and says, you know what? Everybody got flu. I'm the only one who didn't get flu. So these resistance instances and what are the genetics of resistance to infections are most often serendipitously discovered. In this case, I think we could, design a therapy that can be transient and that can inspire us. So can we use IgG15 inhibitors to, not get SARS-CoV-2 severe infection? I think yes, if we had them. And we could pop the pill and for a month and we would be fine. But clearly, genetically having that for the whole life is not evolutionarily beneficial.
Okay, thank you. And then in line of our discussion about whether interferon signature is elevated or not in parents that are carriers for these pathogenic mutations. So parents of COPA-8 children basically have normal interferon signature. What has happened? Have you looked into interferon gene expression signature in parents of patients with IgG15 or ESP18 deficiency, which is a more severe type? Is it normal, elevated?
It's completely normal. So there is no haploinsufficiency that we have detected. Having said that, we have only one line of evidence, none in the cell line, none in peripheral blood. The only line of evidence we have of some sort of a leaky penetrance is from a single set of parents from IgG15 deficiency that we have tested that have developed asymptomatic autoantibodies. A panel of autoantibodies. So that's the only evidence, but molecularly we can detect nothing.
And if you do phosphostat-1 assay in PBMC, stimulated PBMCs of his parents, carriers, that's also normal? Or
that we haven't done.
Yeah, the interferon gene expression signature is done just on peripheral blood, right? So, you just look at for cells that are not stimulated, essentially. But if you stimulate the cells, could it be that there is a little bit of evidence for increased type 1 interferon signaling?
Yeah, we haven't done that experiment, so I don't know.
Okay. I think we have another question, and that is from Cecilia Poli for the SPINA. Do you think flow cytometry protein expression for ADA2 could serve diagnostically?
Thanks, Cecilia. That's a very good question. So what we're doing, we only just started using this antibody for flow cytometry the end of last year. So as we've been going along and getting patients referred, we've been trying to correlate data on flow cytometry expression, immunoblot, and then data to enzyme activity. And hopefully when we do more patients, we will be able to answer your question. I think it's looking good, and perhaps it will have a diagnostic role, but we need more robust data and more patients to be able to just definitely say this can move into a diagnostic lab.
Okay, well, I guess that's all that I see in our chat room. So if there are no other questions, I would like to thank our speakers for participating in this discussion. And, well, I wish all of you to enjoy the rest of the meeting today.
Thank you very much.
Thank you, everyone.
Bye.